All right, looks like we're ready to go. Let's pick up where we left off. Now I'm recording this. I hope you can watch this sometime this weekend. I'm just recording this now after we've just finished the previous activity where we just did this. Okay, we just became the water, and the thing I wanted you to notice, water has a lot of potential orientations it can take when you have a water molecule surrounded by other water molecules. If that water molecule is next to a hydrophobic surface, though, it can't turn in that direction. Now, technically, yeah, it can turn in that direction. It's just not as favorable for it to turn in that direction as it is for it to turn to the other waters. So its entropy, when it is next to a hydrophobic surface, is restricted. If you can get it away from that surface into the middle of a crowd of water, then all of a sudden you can have entropy go up for that water. And this solves a big mystery. If you look at two oily things in water, they will actually appear to come together on their own. If you have two oil drops and you bring them together, you don't have to push them together in water. They'll just come together, and so it will look like they are coming together. It looks like entropy is being reversed and the second law is not valid and everything I ever believed is a lie, et cetera, et cetera. No, don't worry about it. Just because you see two things coming together doesn't mean there aren't other things coming apart and spreading out. So the two things that you're looking at are coming together, and the entropy of them might technically be going down. They're not spreading out, they're doing the opposite. But look at those two things. You know that the two oil droplets in water will be surrounded by waters that are like the people on the edges of the classroom. They're people with reduced orientations relative to where they could be if they were released into the middle of the crowd of water. They're circled right here. When the two oil molecules come together, those water molecules, all the water molecules on the surfaces that are now hidden, have been released. Their orientations have gone up, they are more spread out as a result, and therefore the entropy of the system on the right is higher than the entropy of the system on the left. The system is more spread out, you just have to consider all the parts of the system. And the water is so powerfully spread out because it can assume so many more orientations that it actually ends up driving the two things together. Remember that entropy as long as the entropy of the system increases, what you see happening can happen. In a sense, if you have these 5 water molecules, really small oil drops here, but you have 5 water molecules that were restricted. They were in a solid-like state because they couldn't turn as many directions. When they are released into solution, they become liquid. It's like they melt. And so you have the similar increase of entropy as you do when you're melting 5 molecules of water. Remember that another way to think of it is that when you have the liquid is actually better packed, because the water can actually move around, and those little holes that happen when you first freeze the liquid, those holes are caused by restricted orientations. And so when you release the water, it can actually get closer to other waters, and it actually becomes more dense and better packed. There's multiple ways to look at it, but it's good to think of the water layer being around a nonpolar molecule as being sort of frozen in place. And then it melts when it is released, when the two nonpolar molecules come together and that water can move out. They end up being lower in energy, not because they're forming many more hydrogen bonds and being lower in enthalpy so much as they are higher in entropy. And remember, higher entropy means lower energy. A positive delta S means a negative delta G. All these chemistry terms, and they're coming back to you. And therefore, you will have a situation where if you have oil and water together, they will separate on their own spontaneously. And this will actually involve an increase of entropy. And you have to actually put energy into the system by, say, if you have an Italian dressing bottle, you have to shake it like that, right? You have to shake the bottle to be able to mix the water and oil, and it will spontaneously separate because the water is actually more free by itself than it is in the middle of the oil. And so this happens for 2 oil drops. If you have an oily enzyme and an oily substrate, the same exact thing will happen. This is the same picture as on the last one. It's just showing an oil drop oily substrate, like a waxy substrate and a waxy enzyme. You have that layer of frozen water. When they come together, the water is released, and you actually get a negative delta G. This happens spontaneously, and it happens with a certain energy that pushes it forward. You can actually see the shell of frozen water. That's why they call it frozen. In certain ways of determining protein structure, you can see the waters that are not moving around very much. And you can see they're sort of forced into a high energy shell because they have reduced orientations. They're forming hydrogen bonds, they're just not forming as many hydrogen bonds in as many directions. And so this solves the mystery that you might have had, and actually I had some questions after class of this mystery of how in the world is the entropy of the water increasing when I'm looking at the thing and two things are coming together. Well, it turns out that both things can be true in this way. This is exactly part of the Foundations number 1, page 2 question. I want you to look at this. It's about milk and coagulation. Remember, coagulation is proteins coming together, just like the enzyme and substrate, just like the oily, and proteins have a lot of carbon in them. They have a lot of CH bonds that are exposed, and those are waxy. And so you have all sorts of, you have some parts that can hydrogen bond, but you have some parts that can't. And so proteins, under some conditions, when they are more nonpolar than not, if they don't have many charges on their surface, they will come together by the same kind of nonpolar hydrophobic interaction. We call it hydrophobic because it looks like the waters are afraid of the nonpolar surface. If you let them move away, they will move out from the nonpolar surface and they will be released into solution to a lower energy configuration. There are some parts that this Foundations thing doesn't tell you. Realize that a protein's surface has many of the functional groups that we talked about before. It has several basic functional groups, pKa around 10, and several acidic functional groups, pKa around 4. These are the same functional groups, by the way, that you see in amino acids. So because you know that proteins will have these on their surface, and because you know the pH of lemon juice, if you look it up, is around 2, that will be a titration experiment in which you are changing the charges on the surface of the protein. There, I've given you all the hints for this one. Now I want you to go through and think about how would you explain what's going on with the protein coagulation that you see. And this is actually going to come up again. So this is your introduction to thinking this way. So the thing is, if you have water next to a waxy surface, it will actually, there are some situations in which the water is actually more attracted to itself than it is to the surface. And that can overcome even the force of gravity pulling the water down. For example, you can have something like this. This looks like a bunch of tiles in a bathroom. But what it is it's droplets of water with food coloring in it, like the droplets that you used in your lab this week. But what you have, instead of having it in a well plate, you just have a grid of hydrophobic lines. And the lines are hydrophobic enough, the water is actually repelling from the lines. And it looks like there's an active repulsion force. But realize that, no, actually what it is it's the water that's attracted to itself, and it's attracted to itself so strongly that it's actually forming these. It's just not attracted to the lines at all. Here's a little video of something like this. It looks like a tile, right? But this is water. Oh, let's see if I can get this to actually play. This is a kind of, there we go. That looks like a tile, but it's actually water on a hydrophobic surface. So you want to see that again? I know you can rewind it, but I like to watch it. And you have that going on. The water is binding itself. If you want to talk about where are the bonds are between water. There's just 0 bonds between the water and that surface. So you can make art out of this. There's some artists that have done this, and again, it looks like solid, but it's actually liquid. It's not glass. It is actually water with food coloring. And you can even have a hydrophobic knife that cuts water. So if you look at this right here, this knife is hydrophobic, and this is actually a pretty big drop of water, and it's pretty amazing that you can cut it like a loaf of bread. But you can see that the water deforms and eventually you have enough of the water is holding together that it stays together as a drop, even when you are dividing it with a hydrophobic partition that they call a knife. Okay? This is more information about exactly what it is. It has to do with like Teflon coating and like the Rain X type hydrophobic coating. If you look at those coatings, they all are hydrophobic. They are not able to hydrogen bond effectively with water, and so they are not wettable surfaces. If you've ever used Gore-Tex, you have a hydrophobic surface on that. Gore-Tex is made of many fibers that are woven together, and so they have holes between the fibers. But if you coat the fibers with a hydrophobic surface, you have a really cool effect going on. If you have rain coming down on the surface, the rain will come down in big drops that will splatter, and they'll be scattered by the fibers. And because the surface tension will hold the drops together at a certain size, the drops will have a hard time getting small enough to actually go through. Now, if you know Gore-Tex, it's not exactly a scuba suit. It's not waterproof. If you immerse it in water, the water molecules will push their way through. But if you have droplets, the droplets won't be able to get through. But on the other hand, Gore-Tex can breathe, and water vapor in the gaseous form is really small. It can get out, and it can go up through the holdings of the water. And so you have water that can diffuse out, so you have it being able to breathe, release the moisture from inside, but the moisture can't get in from outside nearly as well. That all comes from a hydrophobic coating around a grid of fibers. And the question of why do some plants look watery because they have just plain hydrophobic waxy surfaces. But there are some plants that actually they have hydrophobic waxy spikes on them. And this can make them look watery as well. If you look at this situation right here, if you zoom in on this, there's actually a water layer being held up, and you've got all this surface tension right here. The surface tension is holding it together so that it's being actually held up by these hydrophobic pillars. It's kind of amazing that the plant can do this, but water's surface tension is high enough that it actually can do this, and it gives it sort of a water layer on top of the fern, and that gives it a silvery sheen. By the way, realize that from this perspective, in a sense, air is hydrophobic. And some of the things that you see, like sea bubbles on the beach bubbles on the grass, on the seashore, they're not on the grass, but you see sea foam and things like that. Those are in part hydrophobic collections that come together because hydrophobic parts will stick out and contact the air. Remember, that's not a positive force between the hydrophobic waxy thing and the air. It's a negative force from the water trying to get away and turning the hydrophobic force, the hydrophobic faces that way. Really complicated, but this is going to keep showing up. We're going to talk about this a lot in the next test unit as well. Cacti actually have the same kind of spikes, and these can actually make water droplets form on them, and then the cacti actually make the water droplets roll down the spikes so that they can be collected by the plant. So the spikes of a different size that form smaller water droplets can actually serve as water collection reservoirs because the water will form a nice hydrogen bonded drop that won't stick to the spike but will roll down it. It's really amazing biomaterials. And so some people have been inspired by this. If a cactus can sort of drink from the air using hydrophobic spikes, can we make a hydrophobic grid that will actually sort of filter out water from the air? And this actually might be viable to give you enough water in the desert that you can set up a hydrophobic grid, the air flows through it, moisture collects, and it drips down into a reservoir. Here's an article about this that you can look up if you're interested, but mostly I want you to have the concept. If you have a hydrophobic surface, water will bead up on it, and then it will collect enough you can use gravity to make the water roll down. Here is a professor at MIT called Evelyn Wang who's actually developed a device. This is a device that will actually collect water from the air, and it will turn it into those little tubes, and you can then drink that water. So I don't know if there's some kind of survival TV show that this might help you on someday. But if you do, I want the credit in some way. I want you just to come back and tell me, thank you for teaching me that leaves can collect water because it allowed me to win Survivor. I don't know. You never know how stuff is going to help you. Here's some stuff about the water-repelling chemistry of what's going on. Remember that CH bonds are hydrophobic. CF bonds are actually also hydrophobic. Those are the coatings that end up in Gore-Tex. The other thing to say how hydrophobic is a surface, how much does the water bead up on it? If the water droplet is flat, then you have a hydrophilic surface. If you have it beading up, you have the hydrophobic surface, and you can measure the angle of the bead to determine how hydrophobic it is. And here's something at the bottom. It might come in useful in Seattle these days. You have urine-repelling wall paint. And so you actually can have, so if you have urine on a wall, I mean, my kids probably, you don't want to go in our bathroom when our kids are little. They get it all over the place. I would kind of like to have some of this stuff because it'd be nice to have the urine actually able to be mopped up rather than to soak into the wall. Hydrophobic coating would allow that. But more important for medical purposes, urine and blood are both hydrophobic are both hydrophilic compounds. They're water solutions. And so if you have a hydrophobic coating, it means blood won't stick to it either. And here's an example of a blood droplet that is on a hydrophobic surface and it's rolling around like a bead. Well, this is really good because usually when you have a medical device, you don't want the patient to stick to the device. You want the stuff to move around. So hydrophobic coatings actually have a purpose for medical devices that you can move bodily fluids around without the bodily fluids sticking to the thing. So, and even could it make a sponge for oil to soak up oil spills? You can actually have a paper like this. So if you have a hydrophobic sponge, the water won't stick to it, but oil will. And wouldn't it be cool to have like an oil sponge? You could just drop it on an oil spill and it would soak up the sponge. It'd soak up into the sponge. So here's the idea right here. This is the hydrophobic group, and just notice looking at this. Yeah, it's got some groups that can hydrogen bond, but really it's got a lot of carbons and hydrogens on it. And if you look around, it's also got this flat surface that metals can actually bind to the pi electrons here. Whatever this is sticky to metal ions. It is sticky to lipids and hydrophobic things, but it is not sticky to water, relatively speaking. So this molecule, if you put it on a sponge and put it into these different solutions of different transition metals, and you can see all the cool colors of the different transition metals, you can see that it's actually going to be able to remove some of the different metal ions as well. So you can have a sponge for metals and a sponge for oils that will leave the water alone. One of the things about this, if it leaves water alone, the water should bead up on it. And this is where they're showing the water contact angle. The water's beading up like this, and it's very hydrophobic. And so if you actually use this, you can actually have a sponge that will actually not soak up water, but it will soak up non-hydrogen bonding materials. And they've tested it with different oils, and you can see that the sponge gains a lot of weight with all these different oils. They're all just different oils. If you look at them, they're like mustard oil and mineral oil. It just looks like that waxy stuff that we showed you before. There's actually a video here, and you know what? You can fast forward through it if this takes up. I meant to have this called up already, but let's just see if I can call it up. Modified sponge soaks up oil but not water. Ah, there it is. So check this out. This is it in full view. There's the sponge, and there's a little droplet of oil, and you see that there it is. It's just soaked up the oil, and it leaves the water alone. So it has hydrophobic and oleophilic. That means oil-loving, okay? But those are kind of the same thing. So you start with a regular sponge, and you give it a hydrophobic coating. Okay, so they take it and they cover it with cyclohexyl groups, which are just a bunch of CH2s over and over again. And here you see the side chains. They make the side chains, all the R's there are cyclohexyls. They call that compound 1. And so it will absorb oils, and you saw that it will also absorb metals. So there's mustard oil and water. And they soaks up the mustard oil, and then you can take it out and the water is clean. If you're able to use a vacuum, you can use it more. I'm not sure how accurate this is. This is good in the lab. Look at it. It's just like pulling up the oil and leaving the water behind. I just think that's really cool. But remember, the oil is attracted to the sponge. The water is attracted to itself. Water is kind of narcissistic, right? So, and there you go. You have some other, this is not a new idea, but this idea is a simple idea. And so you can have the scientist who's talking about it. Okay, you can go back and probably check out the rest of this video another time, but I wanted to show you that in real life. There's really only one more topic that I really want to talk about, and it's going to be the topic of how this works in life. But before I get to that, I want to talk about, it's important that we use, I say a hydrophobic interaction. You know, I was going to say, don't take me seriously. Well, yeah, do take me seriously. Just take me metaphorically. Because when you're talking about a hydrophobic interaction, you're talking about the fact that two oily things come together in water and form a stable structure. But it's really the water that is interacting. So the hydrophobic parts, technically they're not interacting with each other so much as they're allowing the water to interact with itself better. Now, it's okay as a shortcut to say that the two oil droplets are attracted to each other in water. That's fine. That's the way it looks. Just realize if you really want to take it apart, that wouldn't explain why when you measure the entropy of that coming together, the entropy actually increases significantly. You have to account for the water to understand that. Entropy can favor oily things coming together if they come together in a water solution, because the water can pay the price. The guy who first figured this out actually had an interesting life. His name was Henry Frank, and he lived about, I guess it's about 60 years ago now when he made this discovery. And that was during the time of World War II. He was actually a Christian missionary in China who was teaching chemistry. Yes, you can have chemistry missionaries. Apparently this is what he was doing, because teaching about God's world is part of the teaching about God. And so he was teaching about chemistry. He had a PhD in physical chemistry. But if you're in China when World War II started as an American, you're kind of in a bad place. Because after Pearl Harbor, we were at war with Japan, and Japan was at war with China. And eventually he was taken prisoner by Japan. So he was traded for other prisoners. In 1942, he was able to come back to America. And so there he had a teaching job because a lot of the other professors were working on war technology. And maybe he was even a conscientious objector. I'm not really sure about why he wasn't working on that, but he was still teaching because that's what he was called to do. And so he took the place of the permanent faculty who were doing war research, and he taught the students. And in between, he started thinking about this because he was aware of the mystery of entropy increases when oily things come together. And he started to come up with this idea of, well, water's doing this, and there's some evidence that water forms these iceberg structures next to oily surfaces. And so he realized that if that water was released, that would increase its entropy. And so he came up with the interpretation that allowed us to explain the data saying that entropy was increasing. It took about 20 years for people to realize that this teacher, you know, he wasn't the high-powered researcher. He was teaching and doing the research in between his teaching. And it took 20 years for people to realize, oh, this really explains a lot. Let's use this idea over and over again. So that's a good example of how an idea can kind of be ahead of its time, and explaining something can take a long time to percolate through and to be really proved in science. I also wonder, you know, certainly his faith didn't get in the way of him becoming the first person to have this good idea. I wonder if there's any way in which it helped him. Certainly the fact that he was thinking about teaching, when you're teaching, you're thinking stuff through. And as I'm talking to you, I'm thinking about it. And I think that that actually helps you to put together ideas in a better way sometimes. Who knows? Mysterious ways. So the last topic I want to talk about is, okay, when you have oil and water, oil will separate out. What about when you have a molecule that is part oily and part hydrophilic, part charged, okay? So when you have a molecule like that, you have a lipid in the case of life, or you have a soap molecule if you have like an industrial application of that. You have a detergent. In both cases, detergent is just a carbon chain like that waxy chain we showed you, but instead of having another carbon chain stuck onto the ester part, it just has that carbon chain cut off, so you have a fully charged acidic group on one end. You have an example like right here. In water, you have the hydrophilic head group, which has good positive interactions with water that is around it. So the water is interacting with this charge, but then you have this large uncharged surface where the hydrophobic waxy tail is. So there's a water shell around this. By the way, just a little technical thing. There is also a water shell around the hydrophilic head group, but this water shell is not held in place. It can actually reorient itself in many ways because it's got charges that it can turn towards on that surface. So you actually do have a water shell in both places, but the water shell around here, around the lipid part, is frozen in place, whereas the water shell around the head group can turn in many directions, and it has high entropy and is forming good bonds, good plus-minus bonds, which means it has favorable enthalpy as well. So we don't have to worry about that water. That water is good. Now it turns out that this long lipid tail has iceberg water around it, and if you hide those lipid tails, like if you have 3 lipids in 3 of these fatty acid type molecules together, then if you bring 3 of them together like this, look, you've just released all the water at these surfaces that are between them right now. And so you have, this is 3 lipids coming together, but they're drawn together by their by the positive interaction of entropy of water being released, hydrophobic water. Realize that at the same time, we're talking about 3 negative charges being brought really close to each other. So with complex molecules, you have complex interactions, and you have positive and negative interactions. If the tail is long enough, the positive interaction, the positive spontaneity that you get from the things, the tails coming together, the water released and entropy increasing from that, overrides the electrostatic repulsion from the negative heads being brought together, okay? The good news is, I just want you to know that you can have these things going together, and the important thing is if you have a long enough tail, you end up having enough of an attractive force that it overcomes the repulsive forces of the heads coming together. The bottom line is, know that this happens. And if I tell you that this happens, understand why. Hydrophobic effect. And so those come together and they are stable, or in general chemistry terms, they are happy. Okay? Negative delta G if you want to use physical chemistry terms. And so this is one of the reasons why if you have a detergent, the detergent will be sort of, will form structures by the detergent molecules will come together. Detergent molecules look like those fatty acid molecules, long waxy tails, and they'll come together with hydrophobic orientations. Soap films will actually come together with the hydrophobic tails pointing out in some ways. But the whole thing is when you have soap, it makes circles in air. If you have soap in water, it will make little tiny circles that are filled with water. So this is one of the circles it will make. If you have a bunch of those tails coming together, if you have enough of them coming together to form an entire sphere, you actually will have a little micelle is what it's called, a little sphere with an oily inside and a charged group, acidic outside in this case. And if you have a micelle, this is actually a pretty stable structure because all those hydrophobic waters have been released. All of the iceberg waters are now free. Entropy will actually favor micelle formation. The other thing that you'll see happen is as you add more and more detergent, you'll get to a configuration where you have bigger bubbles than the micelle itself will account for. These turn out to be bubbles with water inside, and then they have like a hydrophobic shell around them. Well, how in the world can this happen? This can happen if you have something like this, where you have the hydrophobic shell pointing out, and then below that you have lipids with their tails pointing up, and the hydrophilic heads on the bottom. If you have a layer like this, you can actually have a hydrophobic layer forming, and inside you can have a water bubble. And so you can form a complex structure. Inside that, you can form another little bubble of membrane. And this looks like a live cell. But this can come together just with hydrophobic simple fatty acids in water can form complex dynamic structures. They're not really doing all that much work. What's really happening is the water is sort of alive in the way that it bonds itself dynamically. And the water is moving around so much, it's making the fatty acids move around in a certain way as well. And so the hydrophobic effect produces these very lifelike structures. But this structure right here is just simple molecules in water. And what's enlivening it is the dynamic, dancing structure of water. This looks like a cell. You can have simple molecules that come together in water to form cell-like structures. Is this a way in which sort of lifelike chemistry is baked into the universe? Is it possible that the principle of water forming these complex structures just automatically and spontaneously, does that allow life to emerge from simple molecules? I would used to say, no, it's just too complex. But when I see the complex structures that can form from chemical simplicity, I start to wonder and realize that actually for a long time people thought that it was very natural for life to be born out of non-life. Spontaneous generation would happen a lot in previous theologies. And so the fact that we can think about spontaneous generation happening once upon a time, well, maybe the conditions were right for it, and maybe there's something in the way chemistry comes together that tilts the table toward complex dynamic structures that can give form to life. Either way, it's still God's creation, is the important thing to say. And so this is an idea that the orderly part of the universe where you can have simple things building up into more complex things, that is a seed that can result in reason. And St. Augustine proposed that God created not by making everything like we see it now, but he created by making everything in a previous simpler form, and the whole creation grew up like an embryo growing up into a child and into an adult. That sounds an awful lot like the process of evolution. And St. Augustine had that kind of picture from looking at Genesis 1. Now, there's some ways in which he didn't have that kind of picture, and he didn't say things that went with the science. But I'm more intrigued by the positive resonances I can find, and I think that this theology can actually apply even to chemistry, even at the origins of life. I'm not sure, no one's sure, but I think it's fun to talk about, and it's pretty amazing to see what water can do. So you can consider the hydrophobic rules to be seeds of reason that ultimately leads to life and leads to us. And what's interesting about these hydrophobic rules, seeds store all of their oils in little lipid droplets, and they don't have to push those droplets together. They just let them form because the hydrophobic forces, and remember even that is a little bit of a metaphor, right? But the hydrophobic forces pull them together spontaneously. So lipid droplets allow seeds to store energy spontaneously, and at the same time, could that be an example of a seed of reason that eventually was a principle, a chemical principle that led to order in the universe and a brain that could understand that order and could look beyond that order to the lawgiver of that order. Okay, so it's my birthday, I'm getting theological, sorry about that. But you have that example, and I just think this water stuff is cool. Just a final few examples about how life can have these dynamic, hydrophobic, hydrophilic type droplet structures that are important. If you look at this Vimeo right here, if you just mix fluorescent highlighters in water, so like, I'm sorry, this one's with light sticks, and you mix it together with base, look and see what that looks like, okay? And this has a little bit of music, so because you can't have dancing without music. Or I think it has music. So this is what happens if you take these simple glowing hydrophobic molecules and you mix them together with base. They look like they're alive. That's because the hydrophobic forces are part of what it takes to be alive, and the dynamic structure of water is lively. Okay, I'm going to put it that way. Water is not alive, and the universe is not alive, but the universe is lively. Let me close that just in case because of the wonderful autoplay parts of YouTube. So the last thing I want to show you is I want to show you the some things that talk about this new field of droplet biochemistry. Okay. The physical phase separations, physical hydrophobic effects, will actually control very complex structures. This is a paper that this is an actual paper that talks about this specifically. There are some reviews that have this nice picture. You can see the droplets taking shape, and the droplets give it a colorful, dynamic interior for the cells. These are called phase separations because you kind of have different lipid droplets. It's kind of like oil and water, and they're different phases chemically. And so you have the physical forces, like the hydrophobic forces in water, that are pulling together and form organized droplets. But there's no little person making them come together. What makes them come together is water's H2O structure. This can even result in signaling at neuron membranes. Okay, so we're going to talk about these hydrophobic forces quite a lot in this class. Here's just an advanced example that shows that there is actually a protein called mTORC1 that its job is to control phase separation. If the cell doesn't have this, it gets all messed up. And this protein goes around, it's like, kind of like a shepherd for these little lipid droplets. And it helps the droplets come together. That helps, when the droplets come together, the ribosomes work right. Ribosomes depend on having little lipid-like droplets inside the cell that give the cell a particular structure that allows the cell to function. If you have a structure that is dynamic and can change, you have the potential for homeostasis, and you have the potential for life. All right, so that is my birthday. So thank you for listening to my half-hour theological tinging discussion about the hydrophobic effect. Now work it out for yourself as you work through these homework problems. Work through them iteratively. You're allowed to go back and forth on all of these. They aren't due until the test, but hopefully you'll be working through these this weekend, because on Monday, we're going to take it to the next level. All right, so I will see you then for that, and hope you have a good weekend in the meantime. And thank you for singing today.